Good morning, everyone. So, you must have heard a lot about, you know, uh, bacteria on and inside living at various parts inside our body, whether it is mouth, stomach, small intestine, large intestine, vagina, skin, scalp, everywhere, right? And we have been trying to label them as good and bad, right? And on that basis, we determine what bacteria has to be there and what bacteria should not be there. And this is the biggest flaw in evaluating your biology because, and the fact is, we are still too much obsessed with DNA, whether it is a human DNA or a microbial DNA, right? So DNA is a static molecule, it does not change over life, it remains the same, whether you're healthy, whether you're sick, it is not going to change, right? So what changes is that those genes which are form of the DNA, actually the expression of those genes changes, right? And that's what we determine. Similarly, you know, uh, same microbe, and it's specifically in the context of, a lot of these probiotics which have come up in. So you may say this bug is good, this bacteria is good, this bacteria is bad, right? We focus on that, but essentially there are two things which is important to understand. These bacteria, first thing, living inside your body doesn't mean they will be doing something on their own. They need to be fed with the right fuel to do things, right? So even you're taking certain probiotics, right? Assuming that it will overcome your bloating, constipation, help you lose weight, and a lot of these symptoms, but essentially these bacteria have to be fed with the right things, right? And the name of bacteria really does not matter, right? So what matters is what they are uh, producing, what they are expressing inside the body, right? So again, if you see in a layman's language, our immune cells does not have eyes and ears. They do not understand the name. What they understand is the chemical signals, biochemical activities happening inside the body, and that's why our same bacteria, which is giving good things in one individual, may be giving bad things in another individual. In simple words, one particular bacteria can produce good chemical, anti-inflammatory chemical, pro-inflammatory chemical, both or none, right? It depends upon, again, it doesn't just on its own, it depends upon what you are feeding them. So essentially, as I've shared earlier in my previous videos, also the food is again a sack of 25,000 plus molecules. So when these molecules hit your microbiome, again, 99% genes inside our body belong to these microbes, right? Right. So a lot of these activity inside the body is done by these microbiome, and when it hits your microbiome, a lot of these bacteria interact with these molecules, metabolize them, synthesize them into good thing and bad thing. So the mechanism is, when the food interacts with the microbiome, it can either lead to expression of certain genes, which are good genes, or virulent genes, right? It's a virulent gene, it actually triggers the production of bad chemicals or pro-inflammatory molecules, which overactivates your immune system, stimulates your immune system, then you have inflammation, and that's the starting point of various, you know, uh, chronic diseases, right? Uh, again, it all depends upon the expression of the genes, right? So we all know that microbes outpower human genes by 150 to 1, but not all genes are being expressed at one point in time, and we don't need to understand all genes. What we need to understand, only few of those genes, or out of the 2 million genes, maybe only 80,000 or 100,000 genes must be expressed, right? So that is something which is really important to understand. So what you eat, right? And based on that, what metabolites your microbiome are producing, right? As a result of what genes are being expressed is what matters. So in simple words, food you eat goes to your microbiome, your microbes metabolize it into various molecules which can be good and bad. And what we really need to understand is what the so even you're taking sorry, you're taking probiotics, you need to understand that which pathway or which metabolite or which chemical this bacteria is going to produce, right? If it enters my body, giving your current dietary choices, right? So in simple words, the bacteria can be good and bad, or both or none, right? So it all depends. It does not depends upon which bacteria is there. It depends upon what they are doing, what they are producing, right? So in simple words, what we do is, so when we again, we do not focus on DNA, we do not focus on, understanding which bacteria is there, right? Which genes are there. We just focus more on, which genes are being expressed, what functions are being performed by these microbiome. So when we take your biological sample, suppose we're taking your gut microbiome, we take your poop sample, there are, millions of molecular information in the sample, right? It goes through our obviously the sequencing process. When we sequence these molecules, right, we have a lot of this raw data which you and me cannot understand. So it goes to our, you know, bioinformatics algorithm, where it is actually converted into, genes expression data, where we get from 130 bytes of molecule 130 million bytes of molecular information, we convert into 100,000 genes expression. Again, these genes expression, again, as the next step, based on our mathematical model, we actually classify and reclassify these genes expression into various pathways, right? And the probiotics, if you are taking, or the food which you have to take, depends upon the functional activity level of each of these pathways. We quantify and measure and quantify each of these pathways, right? So you can even understand, if I'm going to, take a particular, for instance, E. coli, if it's a bacteria, if I'm going to feed it, is it going to give me vitamin B12, or it is going to trigger inflammation, trigger the production of LPS, right? Similarly, the probiotics, decision to take a particular strain of probiotic and what quantity depends upon the quantification of these pathways, right? So all these probiotics based on, you know, labeling as good and bad bacteria is not the right approach, right? We really need to understand which metabolites are being produced as a result of interaction between food and the particular microbiome, and that is what would drive your nutrition intervention, your supplement intervention, your prebiotics and probiotics intervention. Right? That is really, really important. Do not fall for such probiotics, which does not talk about what metabolite it is going to produce, what strain you are being fed with, not species. You really need to understand the strain levels. So just to sum up, there's nothing called as good and bad bacteria. Bacteria can be good, bad, none or both, depending upon, what genes, what microbial genes are being expressed, what metabolites are being produced, right? And what you're feeding them with. Right? That's it for today. Have a great day. Thank you very much.